Hello, my name is John Jacque. I'm a senior scientific cloud consultant at Bioteam. Today, I'll be speaking on the topic of utilizing generative AI techniques for drug discovery and therapeutic development. Before diving into these techniques, I wanted to touch on an important concept: pragmatic AI. Pragmatic AI is how Bioteam approaches problems utilizing artificial intelligence and machine learning technology. So, what do we mean by pragmatic AI? Simply put, it's a practical, realistic approach to artificial intelligence. It's a concrete, well defined, domain specific application that cuts through the hype, accelerating the process of solving highly specific, real world scientific or business problems using the tools we have available right now. This method helps you minimize the risks of AI adoption, and most importantly, it allows you to achieve measurable results in key areas like scientific discovery, process automation, or the automation of scientific discovery. We also have to address the elephant in the room, the LLM bubble. If it bursts, I believe these domain-specific applications and techniques we present will stand the test of time. So here's a great example of the pragmatic application of AI technology: computational chemistry and drug discovery pipelines. Traditionally, determining the structure of a protein has been extremely difficult, time-consuming, specialized, and very resource-intensive. It's also an extremely important task because the shape of a protein or molecule informs its behavior and function. It's almost a one-to-one relationship. That's where AI comes in, with protein structure prediction models. These models take sequences of amino acids and predicts the shapes they will fold into as a protein. Running inference on these models can buy down your time to discovery from weeks, months, and years to mere hours or days, revolutionizing the therapeutic discovery process. Though it is important to note, the wet lab processes and approaches will always be important for validation, though AI can augment and accelerate the discovery process. In addition to predicting protein structure, these models can also help predict biomolecular properties: thermal stability, binding affinity, hydrophobicity, to name a few. And these models work great in isolation, but they can also be wired together and composed into larger, higher order scientific discovery pipelines. You could think of it like building a Lego set. The individual bricks are the predictive AI models, and the fully assembled set is your discovery pipeline. For example, RF antibody, which is used to design and rank novel antibodies that bind to given antigens. This goes beyond the discrete capabilities of AlphaFold. Now, it's worth noting that RF antibody was a pipeline that Bioteam helped port to the cloud using a service called HealthOmics in partnership with AWS. So hopefully by this point, I've conveyed the power, composability, and potential of these models in accelerating the scientific discovery process. If you're sold on the idea, your next question might be, how can I get started running these models and pipelines? The answer is, you need your infrastructure to be AI ready. So what makes infrastructure AI ready? It means your infrastructure needs to do a lot of math really, really fast, specifically matrix multiplication operations. And it needs to do these operations in parallel very, very quickly. Traditional general-purpose CPUs are ineffective for this task. They simply weren't designed for it. That's why auxiliary purpose-built accelerators are needed. You also need high-speed memory, a lot of PCIe bandwidth to shuttle data in and out of the accelerators. Sitting on top of this hardware, you need the computing platform software, including drivers, middleware, and libraries. Now, these AI workloads are inherently spiky. They require massive amounts of compute in the form of teraflops and fast, expensive memory. Some of these models run for minutes, others hours. When purchasing the hardware to run these workloads, any idle time could be considered waste or an inefficient use of resources. There's also a prohibitive latency for scaling beyond your current capacity in the hardware procurement process. On the flip side, if you're considering renting your compute from a cloud service provider, running a known and well-defined workload volume could be more expensive than necessary. Wouldn't it be nice to not have to make a choice, to do both as needed when the underlying economics and market changes? Enter infrastructure decoupled workloads. These are workloads that are agnostic to the underlying compute platform or service that it runs on. Workflow orchestration languages like Nextflow and Whittle, combined with containerization tools, can help you abstract your workloads and decouple them from the infrastructure layer, enabling flexibility and choice. This diagram you see to the right illustrates a spectrum of considerations you can use when selecting where your workload executes. Do you have a known or unknown run volume? Are you working with a workload that's already operating in production, or is it more of an unproven prototype? Have you or are you able to make capital investments in hardware? Will in-house expertise be able to support the level of ownership required? By answering these questions, you can align your workloads to the infrastructure best suited for your organization. And this is something Bioteam can help with. While this diagram may be pretty and symmetrical, there are many nuances to consider. Nuances that map to an n-dimensional space beyond the single dimension continuum illustrated here. Now, in the spirit of open source, we'd also like to share a few models with you. Bioteam has ported CompChem AI models and workflows to the HealthOmics service in tandem with AWS. You can get started in minutes with end-to-end inference working in a matter of hours. We'll provide this QR code again later at the end of the lightning talk. And that's it. Thanks so much for watching and listening. We hope you'll stay in touch.